Friends, welcome to Biology Made Easy. Today in this video, we'll discuss lysosome. So lysosome is an organelle that was discovered by the Belgian scientist Christian de Duve in 1955. Christian de Duve in 1955 we discovered this, but this was discovered not by observation through microscope, only through different biochemical analysis, but through electron microscope it was observed in the next year by Novikov in 1956. Now this lysosome, it is present in most of the eukaryotic cells and mostly only animal cells. They are not present in plant cells. So they are present in animal cells except RBC. They are also present in fungi and some protozoans, but they are absent in the plants, and they are abundantly present in white blood corpuscles, macrophages, then Kupffer cell, then pancreas, spleen, liver, and kidney. So in these cases, the lysosomes are abundantly present, while they are absent in all these cases. Now, what is the structure, function, all these things let us discuss. Usually, the lysosome is produced from the Golgi bodies through the jaw system that we have discussed in the previous video, where we have discussed how the jaw system functions. That means the endoplasmic reticulum gives rise to Golgi body, and the Golgi body gives rise to lysosomes. So usually the lysosomes arise from Golgi body through jaw system, Golgi body, endoplasmic and lysosome from this through this jaw system. So what is the structure? Usually the lysosome is a spherical structure, or you can tell round structure, and having the diameter about 0.2 to 0.8 micrometer. And this is only a single membrane structure, and this single membrane consists of that is unit membrane, single membrane, and also it is a unit membrane. That means like mitochondria and chloroplast, it is not double membrane, single membrane structure. Now inside this lysosome, which is spherical structure and present in animal cells, around 50 digestive enzymes are present. Around 50 digestive enzymes. So about 50 digestive enzymes are present, and these digestive enzymes, they are phosphatases, sulfatases, proteases, lipases, amylases, like phosphatases, sulfatases, nucleases, proteases, peptidases, and then lipases, amylases. Likewise, 50 enzymes are present, and all these enzymes are mostly the digestive enzymes, and all these enzymes together known as acid hydrolysis. This is very important from examination point of view, acid hydrolysis. That means the acid hydrolysis are the marker enzymes of this lysosome. Now, one important thing is all these acid hydrolyses, they function only in acidic pH. That means they function in acidic pH, and the pH ranges usually from 4.6 to 5. Now the question is how this pH is maintained. It is maintained due to pumping of protons, which is ATP driven or energy driven. So how the whole thing takes place? Now this is the ATPase enzyme, and now one molecule of ATP will break down to ADP and inorganic phosphate, and it will release the energy. Now when it will release energy, that energy will be utilized in pumping of protons to this lysosome. That means always this lysosome, in the lysosome there will be pumping of protons due to expenditure of energy, so that this pH is always maintained, and in this pH only all these enzymes are functional. Now you may ask one question: if all these are digestive enzymes present inside the lysosome, then why it is not digesting the lysosomal membrane itself? Actually, it's a very good question. So what is the answer to it? Usually this unit membrane which is present in the lysosome, it protects itself from the enzymes of the internal enzymes, because it is its proteins and lipids are highly glycosylated. That means the proteins and lipids of membrane are highly glycosylated. That is why the enzymes cannot digest it. Then under what condition they will be digested? That means this lysosomal membrane will be fragile. It will break only when there will be presence of certain membrane labilizers. So what are these labilizers? Only in the presence of this membrane labilizers, the membrane can break down. So the membrane labilizers, are usually absence of oxygen. That means if the oxygen concentration will be low, then there is possibility. Then excess of vitamin A and vitamin E. Then sex hormones. In addition to that, also the X-ray, then UV light, then detergents, then bacterial toxins, then bile salts. So these are known as membrane labilizers. That means if these will be present, then that will the membrane will be labilized, that will become fragile, and the enzymes will come out. Now, in addition to this glycosylated lipids and proteins, there are also certain other substances which stabilize that protein, and they are known as membrane stabilizers. Labilizers and stabilizers. So these are usually cortisol, cholesterol, then that is quinine and heparin. These are the substances which are usually known as stabilizers. That means if these substances are present in the cell, that will stabilize the membrane in addition to this glycosylation, and while if these are present, then they will make the lysosomal membrane fragile and lysis. Now the next thing that we have to discuss is this lysosome is of different types. That means it shows polymorphism. That means lysosome is of 4 different types, and they are usually known as primary lysosomes, then secondary lysosomes, residual bodies or tertiary lysosomes, and autophagy vacuoles or cytolysosomes. So the next thing that we'll discuss is that is the polymorphism. Polymorphism, or you can tell that is the types, polymorphism or types. So usually it is of 4 different types. 4 different types. Now the number 1 is primary lysosome. Primary lysosome, which is also known as storage granule. Storage granule. So when the lysosome is pitched up from the Golgi body, that is known as primary lysosome. That means it is pinched off from Golgi body, and it contains some enzymes which are inactive. Contains inactive enzymes. That is why it is also known as storage granule. Now the second one. The second one is known as secondary lysosome. Secondary lysosome, or it is also known as heterophagosome. Heterophagosome or phagolysosome, or also it is known as the digestive vacuole. These are the different names. So how this secondary lysosome or heterophagosome is formed? Usually to the cell, the solid food materials, they come in the form of phagosome. When the phagosome, which is loaded with food materials, combines with the primary lysosome having inactive proteins, it gives rise to secondary lysosome. That means secondary lysosome is the combination of a primary lysosome plus phagosome loaded with food materials. So when the primary lysosome combines with phagosome to give rise to secondary lysosome, naturally function is digestion. So the digested substances will be absorbed and it will enter into the cytoplasm, while the undigested food material that will remain in the rest of the lysosome. That is, since it is digestive food material, that is why it is also known as digestive vacuole, and this process is also known as heterophagy. This process is known as heterophagy. By this method, not only the phagosomes are digested, even the pathogens like bacteria, virus, or any other pathogen that attacks the cell, they can be digested by this method. That provides a defense to our body. So through this secondary lysosome, they are digested. Now let us discuss the third type. The third type is known as residual body. Residual body, or known as tertiary lysosome. Tertiary lysosome, or also it is known as telolysosome. Telolysosome. Now what is this residual body? After this secondary lysosome digests the food material, the digested food material will enter into the cytoplasm. Now the undigested substances which remains in the lysosome, that is now known as the residual body. So what is the function of the residual body since it is undigested? Now this residual body which contains undigested food material, that will pass towards outside, towards the cell membrane, and by means of exocytosis, that undigested food material will be removed out of the cell. That means it will be removed by exocytosis. By exocytosis, it is removed. And this process is also known as ephagy. This process is known as ephagy. This to exocytosis it is removed. But somehow, if the exocytosis cannot take place, the undigested substance cannot be removed from the cell. That means it will be stored inside the cell. That will lead to some pathological diseases, which are also known as storage diseases. That means the accumulation of residual bodies will lead to pathological diseases, or which are also known as storage diseases. So what are these storage diseases? Now usually it is hepatitis, polynephritis, then Tay-Sachs disease, then Hurl's disease, also Pompe's disease. All these diseases are known as pathological diseases or storage diseases. That means due to accumulation of the residual bodies. Again, due to accumulation of this residual body, it also gives rise to the lipoprotein granules of the nerve cells. They are nothing. The lipoprotein granules of the nerve cells, they are only the residual bodies. So the lipoprotein granules of nerve cells are also residual bodies. So this is about the third one, that is the residual body. Now the fourth type, there is also fourth type of this lysosome, and that is known as autophagic vacuole, or that is also known as cytolysosome. The fourth type is autophagic vacuole, or it is also known as cytolysosome. Now this autophagic vacuole or cytolysosome, it has two important functions. One is autophagy, the other is autolysis. It has two functions. One is autophagy and the other is autolysis. Now these two are also very important. Now what is autophagy? Sometimes in the cell there are certain organelles which degenerate, or they may be surplus, or they may be defunct, and their presence inside the cell is not necessary. They should be destroyed. So in that case, so what will happen? In that case, suppose this is one organelle which is defunct or it extra surplus. So in this case, this will be surrounded by the lysosomes. It is surrounded by lysosomes and completely it is digested. So when it is completely digested and disposed of, so this is known as also apoptosis. We can call it apoptosis. And since this degenerated and extra organelle that is disposed of, we can call it as a disposal unit. Further, when these substances are disposed of, then they can be reutilized for synthesis of other substances. That means these can work as recycling centers. That means due to the function of autophagy, it leads to apoptosis. It acts as a disposal unit and it acts as a recycling center, because those organelles which are surplus or extra, they are completely disposed of due to the action. This is known as autophagy. Now what is autolysis? Lysis means breakdown, auto means self. That means in this case autophagy, what happens? the extra or surplus organelle is surrounded by the lysosomes and digest it. But in case of autolysis, the lysosomal membrane, due to presence of the labilizers or something, that will completely break down and all enzymes will come out and they will digest all the parts of the cell. That is known as autolysis. That means here the membrane breaks and enzymes come out. As a result, what happens? The cell, tissue, they are all digested. Now, in which case all these things happen? When these enzymes will come out, they will digest. Usually, this is done in case of aging cells, dead cells, and disease cells. That means these cells which are not functioning any further. In that case, the membrane will break, enzymes will come out, and they will digest this cell or tissue, mostly which are aged cells, they are dead cells, or they are diseased cells. Now, this thing happens mostly when there is a metamorphosis of a frog. So, when metamorphosis takes place, the tail is removed, that is digested. So, the metamorphosis of a frog, that is one example. The extra organelles, they are completely digested by this method. Now, because in this case, the enzymes are coming out and they are digesting the cell, tissue, that is why the lysosomes are known as suicidal bags of the cell. For this reason, they are known as suicidal bag of the cell. So, this is the fourth type, that is autophagy vacuole. Now, what is autophagy and what is autolysis, clear. Now, let us discuss functions. Functions already we have discussed because all the functions are included here, but if we summarize them, then we can write it here in a summary manner. What are the different types of functions? Now, one is intracellular digestion. That means the food material present inside the cell, they can be digested by lysosomes because it contains around 50 digestive enzymes. Similarly, the extracellular digestion, which is known as heterophagy. Then the self-defense. Now, this due to heterophagy, when the pathogens attack, they come through the phagosomes, so that can be digested. That means the cell protects itself. That is, the lysosomal functions are for self-defense. Then they function as scavengers. How? The residual body, they remove the waste materials. So they act as scavengers. Here you can write residual bodies. Then they act as disposal units. Because here we have seen that the extras or surplus one is digested and they are disposed of. So they work as disposal units. They work as also recycling centers, because the disposed ones, they can be recycled again. They are also responsible for production of thyroxine in thyroid. And another function is they help in fertilization. The lysosomes which are present in the sperm, they help in digesting the sheath of the egg. So they aid in fertilization, and sometimes also they initiate cell division, that is mitosis, by digesting the repressors, the inhibitors, help in mitosis. So these are the different types of the lysosomes, that is polymorphism and the different functions. So this lysosome is very important from examination point of view. They are present only in case of animal cells, and some fungi they are not present in case of plants. Then you may ask, if they are not present in plants, how this digestive enzyme, where they are present? In case of plants, the plant lysosome, usually the spirosome in case of plants. In case of plants, the spirosome, the aileron grains, and vacuoles, they function as the lysosome in case of plants. That means most of the enzymes they are present in the vacuoles, the aileron grains, and spirosome. So these three, they are known as the plant lysosomes, because that lysosome is not present in case of plants. So now you have a clear concept about the lysosomes, the types and the functions. Thank you very much for watching the video. Wish you a very bright future. God bless you.